James Webb found something it was not supposed to find. In the atmosphere of a distant ocean world, 124 light-years from Earth, the telescope detected the chemical fingerprint of a molecule that, on this planet, is produced by one thing only: life. The team that found it announced it. The scientific press ran with it. For a few months, the question felt settled. Then other teams asked for the same data. They ran their own analysis. They found something different. The signal was there, and then it was not. That is where the story begins. In September 2023, a team led by Niku Madhusudan at the University of Cambridge published a paper in the Astrophysical Journal Letters. Using observations from the James Webb Space Telescope, they had analyzed the atmosphere of a planet called K2-18b, a world orbiting a dim red dwarf star in the constellation Leo Minor. The paper reported something no telescope had managed before. For the first time, carbon-bearing molecules, methane and carbon dioxide, had been detected in the atmosphere of a planet sitting inside its star's habitable zone. The European Space Agency called it a step toward finding life elsewhere. The phrase floated through headlines for weeks. The planet itself is not Earth. It is nothing like Earth. K2-18b is a sub-Neptune, 8.6 times the mass of our world, 2.6 times its width, a category of planet that does not exist anywhere in our own solar system. At that size, the models say it likely holds a global ocean, not a lake, not a sea, but a world-spanning body of water with no shore, no continent, no boundary, beneath a sky of hydrogen and helium, at a temperature around -8 degrees Celsius. The ocean, if it exists, would be cold enough to preserve complex chemistry. The atmosphere above it would trap heat without becoming a pressure furnace. Scientists had a name for this configuration. They called it a Haitian world, a portmanteau of hydrogen and ocean, and K2-18b was their best candidate. Think about what that means in terms of scale. If that ocean exists, it covers the entire surface of a world larger than any planet in our own solar system's habitable zone. No shore, no mountain range breaking the horizon. Just water, stretching from pole to pole, beneath a hydrogen sky too thick for sunlight to reach the way it reaches us here. And at 124 light-years away, the light Webb is reading right now left K2-18b before the Wright brothers flew, before Einstein published his first major papers, before the First World War began. James Webb is not reading the planet as it is. It is reading what that atmosphere looked like over a century ago. The molecule detections mattered for a specific reason. Methane and carbon dioxide together, in a hydrogen-rich atmosphere, without any detectable ammonia, is not a random combination. That pattern had been proposed as a biosignature pair years before Webb ever launched. The argument went like this: life consumes carbon dioxide and produces methane. The absence of ammonia is consistent with a surface ocean suppressing it. When you see that chemistry in a habitable zone atmosphere, the simplest biological interpretation is that something down there is metabolizing. Webb had aimed at K2-18b, and the chemistry had appeared exactly where the models predicted it should appear if life were present. That should have been the moment everyone exhaled. But it was not, because buried inside the 2023 paper was something smaller, a whisper in the data. The Cambridge team noted a faint spectral feature at a wavelength associated with dimethyl sulfide, DMS, a sulfur-bearing organic molecule. On Earth, DMS is produced by one class of source: marine organisms. Phytoplankton, bacteria in the water, they release it constantly, and it rises through the atmosphere before being destroyed by sunlight. No geological process makes DMS. No volcanic eruption, no rock weathering, just biology, working in the ocean. DMS is also chemically fragile. Once it enters an atmosphere, photochemistry breaks it apart within roughly a day, which means any planet showing sustained DMS would require a continuous biological source actively replacing it, something alive, producing it constantly, at a scale large enough to be measurable from 124 light-years away. The team logged the feature, hedged their language, and moved on. The signal was between 1 and 2.4 sigma, below any standard threshold for a detection. It appeared too weak to claim, too specific to fully dismiss. For the next 18 months, that whisper waited. Then came 2025. The Cambridge team had obtained new JWST observations using a different instrument, MIRI, the mid-infrared camera, designed to read longer wavelengths where different molecules leave different marks. This time, the result was harder to quietly set aside. The MIRI data showed a distinct spectral feature. The team's analysis found it inconsistent with a featureless spectrum at 3.4 sigma confidence. When they tested which molecules could explain the shape of it, most candidates failed. The only ones that consistently fit the data were DMS and dimethyl disulfide, DMDS, a closely related sulfur-bearing molecule also linked to biological chemistry. Independent evidence for one or both, they reported, at 3 sigma. The paper was accepted for publication in the Astrophysical Journal Letters. This was not a tabloid claim. 3 sigma means roughly a 1 in 1,000 probability the signal is random noise, an accepted threshold for reporting a significant scientific finding. The team was careful with their language. They did not write alien life. They did not write proof. They wrote that the evidence was consistent with a biological source, that more observations were needed, that the interpretation required caution. The word life never appeared in the paper, but it did not need to. The media put it there. Within hours, the claim was traveling under headlines the Cambridge team had not written, and in some cases, had not intended. NASA's own communications used language like hints of dimethyl sulfide, and more work is needed to confirm this. Neither NASA nor the European Space Agency called it a biosignature detection. Both agencies hedged carefully, not because they were withholding something, but because neither was willing to make a claim the data could not fully support. The difference between what the agencies said and what the headlines said was not a gap in honesty. It was a gap in standards. That is the first version of this story, and it sounds complete. A world with the right temperature, the right chemistry, a global ocean, and now the specific molecular fingerprint of marine biology, found twice, with the world's most powerful telescope, reported in a peer-reviewed journal. By that reading, K2-18b was the most significant astronomical finding in a generation. You could stop there and feel like the case was answered. Here is why it was not. The MIRI instrument operates in a wavelength range that is genuinely difficult to calibrate. The transit spectrum, the signal extracted as a planet passes in front of its star, is highly sensitive to a technical choice called wavelength binning, how you group the incoming photons before reading them. Change the bins, and the spectrum changes, not because the planet changed, because you changed how you listened to it. Multiple researchers examining the MIRI data noted this immediately. The spectral feature that the Cambridge team identified appeared under their chosen binning scheme. It was not stable across all possible configurations. This was not a mistake. It was a fundamental uncertainty built into the instrument itself at those wavelengths, one that the data, on its own, could not resolve. The full weight of that would not become clear until other teams started working. Within weeks of the April 2025 announcement, independent groups around the world requested access to the publicly available JWST data and began their own analyses, not to undermine, not to challenge for the sake of it. Because that is how science is supposed to move. One team reports a result, and multiple others check it, using different tools, different software, different assumptions. What those teams found was not a confirmation. A study led by Raphael Luque combined the MIRI data with the full near-infrared observations from NIRISS and NIRSPEC and found insufficient evidence for DMS or DMDS. A team led by Kevin Stevenson applied independent retrieval frameworks and reported that the MIRI spectrum was highly susceptible to unresolved instrumental systematics, and that in 87.5% of their retrieval configurations, no DMS signal appeared at all. Not a weaker signal, no signal. The same raw data, the same telescope, the same sky, different pipelines, and completely different conclusions. The Cambridge team did not retract. They stood by their published findings and pointed to the peer review process. They noted that different methodological choices can produce different results, and that deciding which method is correct is itself an open scientific debate. That is a legitimate position. 3 sigma is not invented. The signal appeared in their analysis. It simply did not appear in anyone else's. The question was no longer what the telescope found. The question was which telescope result you were reading and whose method you trusted. The answer, when it came, was consistent. 3 independent teams. A multi-team collaborative study led by Renyu Hu at NASA's Jet Propulsion Laboratory. All of them working from the same public JWST dataset, all applying Bayesian analysis and independent atmospheric retrieval frameworks. The July 2025 Hu study combined 4 new JWST observations with the original data and invited each team to reach its own conclusions independently. The result was convergence, not on the presence of DMS, but on its absence. No conclusive evidence for DMS or DMDS. The features in the MIRI mid-infrared spectrum were in tension with the more robust, smaller features visible in the near-infrared range. Instrumental red noise, rather than atmospheric chemistry, appeared to be the more likely explanation for what the mid-infrared data had been attributed to. That is the 3rd version of this story. And it is about more than one planet, because the debate over K2-18b's DMS signal is also a debate about process. The Cambridge team published a result in a peer-reviewed journal. That is what scientists are supposed to do. What the broader community did next, requesting data, running independent analyses, publishing counter-studies, is also exactly what is supposed to happen. The problem is timing. The announcement traveled at the speed of headlines. The verification traveled at the speed of science, which is considerably slower. By the time multiple teams had concluded the signal was not robust, the story was already embedded in public understanding. K2-18b had alien life, or something close to it. Then, quietly, in preprints and journal papers that received a fraction of the original coverage, the signal dissolved. This is not unique to K2-18b. Astrobiology has watched this cycle before. A result reported with appropriate scientific caution, covered without it, then complicated by follow-up analysis. What is new is that Webb has the power to make that cycle run faster than any telescope before it. This is not a story about NASA hiding something. It is a story about how long it takes a scientific claim to be tested, and how short the distance is between announcing a 3 sigma result and watching the world treat it as certainty. The agencies were careful. The agencies hedged. The gap between what was said and what the public heard was not a failure of disclosure. It was a reflection of how difficult it is to communicate the difference between a finding and a conclusion, and how rarely that distinction survives the first hour of coverage. But here is where the story changes shape. And this is the detail that was not in the DMS announcement, because it had not happened yet. In October 2024, a team analyzing data from the European Space Agency's Rosetta mission published a result that should have rewritten the K2-18b conversation immediately. Rosetta had orbited Comet 67P Churyumov-Gerasimenko, passing instruments through the comet's coma to analyze its chemistry in extraordinary detail. The data from the Rosina mass spectrometer showed something that had never been found before in space: dimethyl sulfide, present in the pristine chemical inventory of a comet. No ocean, no biology, no phytoplankton, just ice, dust, and the chemical reactions that occur in the frozen interior of a body that formed when the solar system was young. Dimethyl sulfide, the molecule that scientists had said nothing except life can produce, was sitting inside a comet. Then, in early 2025, radio astronomers announced that DMS had been detected in the interstellar medium. In a molecular cloud near the galactic center, between stars, in cold gas where no life as we understand it could survive, the molecule was present. It had apparently formed through abiotic chemistry, reactions between simple molecules on icy dust grains, or in the gas phase, no organism required. The researchers were careful to note that DMS formation pathways in the interstellar medium are not yet well understood, and that this did not immediately disqualify DMS as a biosignature in all contexts. But the underlying logic had fractured. DMS was treated as a reliable life indicator precisely because no natural non-biological source had ever been identified. Not on Earth, not anywhere in space. That claim had now broken twice in the same 12 months. The molecule that was supposed to prove life turned out to be everywhere. Life is not. This changes the K2-18b question at a fundamental level. Even if the Cambridge team's signal were real, even if future observations confirmed DMS in that atmosphere at a level no independent team could dispute, the question of what it means would remain open. An atmosphere containing DMS is not automatically an atmosphere shaped by biology. It may be. But the exclusivity that made DMS compelling as forensic evidence is no longer intact. A comet found it. A molecular cloud found it. The chemistry of the interstellar medium, present long before any living world existed, can apparently produce it without a single cell. Consider what the ocean on K2-18b would need to look like if the DMS were biogenic. It would cover the entire planet, pole to pole, no land anywhere, nothing but water stretching to every horizon. The biological system producing DMS would have to be vast enough to maintain the molecule in the atmosphere continuously, fighting the photochemistry that destroys it every single day. On Earth, our oceans produce DMS through billions of tons of marine microorganisms working at the surface. K2-18b's hypothetical ocean would require a comparable biomass, or greater, spread across a world nearly 3 times our diameter, operating beneath a red dwarf star that delivers a fraction of the energy our sun provides. Those organisms would never see a star rise above a horizon. They would drift beneath a hydrogen sky, producing a molecule that rises and gets destroyed, and producing it again, day after day, across an ocean with no edge. They would be the most productive biosphere in known history, invisible to us except as a faint chemical trace in light that is over a century old by the time it reaches our mirror. That picture was never impossible. It is simply no longer required to explain the data. What Webb genuinely found on K2-18b is not nothing. The WHO and others' 2025 collaborative study confirmed methane and carbon dioxide in the atmosphere at robust statistical significance, It found that K2-18b is a water-rich world. The simultaneous presence of methane and carbon dioxide at the observed abundances can only be explained by either a massive atmosphere with a very high water content or a smaller atmosphere overlying a liquid water ocean. The planet is a plausible Hycean world. The chemistry is real. The atmosphere is not inert. And the methane and carbon dioxide pair, without ammonia, remains consistent with what models predict for a biologically active ocean surface, though it is also consistent with certain abiotic scenarios that do not require life at all. None of that vanished when the DMS signal weakened. The planet is still the most chemically detailed habitable zone world JWST has studied. That result stands. But what K2-18b was supposed to become, the confirmation, the first forensic trace of biology beyond Earth, remains out of reach. Not because the planet is wrong, because the tools we are using to read it have limits that we are only now fully beginning to see. That is what makes this uncomfortable. K2-18b represents the best case we have. The best planet, the best telescope, the best instruments, years of repeated observation, multiple independent research groups applying the most sophisticated atmospheric retrieval methods available. And the result is, the signal is uncertain. The instrument is unstable at the relevant wavelengths. The molecule at the center of the claim can apparently form without life. And the standards science requires to call something a discovery, 5 sigma, independent confirmation, results robust across all methods, have not been met. James Webb is still watching K2-18b. More observations are scheduled. The Cambridge team stands by their published findings. The broader community continues to analyze and revise. No side has withdrawn. The data keeps arriving, photon by photon, filtered through an alien sky 124 light-years away, traveling for over a century before it touches our mirror. At that distance, nothing can be staged. Nothing can be added. There is only what the atmosphere of that planet is actually doing, and the question of whether the instruments humans have built are sensitive and stable enough to read it honestly. Somewhere in a molecular cloud between stars, dimethyl sulfide drifts in the cold dark. No ocean beneath it. No life making it. Just chemistry, ice and gas, and reactions that have been occurring in the space between stars since long before Earth existed. It formed there without life. It was found in a comet. It may be forming right now in 100 places no organism has ever touched. K2-18b may still have an answer. The planet has not changed. The light has always been there. We simply do not yet have the tools to read it with enough certainty to know what the answer is. And the honest part of that sentence is that we did not fully understand that until we tried.